Okay, so I said yesterday we didn't have enough time. We'll finish it today, so we'll start doing the drawing right now to kind of show what happens with the sodium and the potassium channels that cause an active potential to occur. While this is going on, this active potential that we're going to talk about, we're going to break it up into different phases. While this is going on, this is generating the active potential. Keep in mind, what pump did we learn about yesterday that controls sodium and potassium concentrations? Sodium, potassium, ATPase pump. Make sure you put that ATPase because you know it's a powered pump. Means it's going against the gradients. So while this is going on, the sodium potassium pump is also occurring to keep these gradients fixed so that we always have potassium wanting to leave the cell, sodium wanting to come inside the cell. So this is set up in 4 different areas. We're going to look at the first one by itself, and phase 1 is the resting phase. At the resting phase, all the gates are closed. So we'll do a blue gate here. That's closed. And a gate there. And remember I said the sodium has an inactivation gate. That little thing that I drew at the bottom is the inactivation gate. And you'll see where this pops in when we get to different phases. Okay? So potassium gates are shut. Oh, sorry, sodium. I'll make sure it's there. So that's shut. Now, let's talk about the concentration of sodium. Where is the sodium mainly found? Outside the cell. So extracellular, we'll just put some circles out here. And these blue circles represent the concentration of sodium. We know it's more outside the cell than inside the cell, so I put more blue dots out there. And inside the cell, I'll just put a couple. So we know that's the direction that sodium is going to want to go. The yellow side, I'll do it in orange, is our potassium channels. These are also closed at this point as well. So we'll put some doors here. Potassium does not have the inactivation gate that sodium does. So we're not going to draw one little funny little hat there because it doesn't exist on the potassium channels. For potassium, where is potassium mainly located? In or out? In. So we'll put some orange circles down here. So we know there's a high concentration of potassium inside, but outside we have a few, or outside there's a few potassium ions there as well. But we've now set up our concentration gradients. We've analyzed the doors. Everything's closed at rest. Now, while we are at rest, what is the resting membrane potential of a cell at rest? Negative 70. So let's go down to this graph all the way down here at the bottom. Phase 1 is resting. Just like we did in phase 1, we drew the boxes before. We're still in phase 1, we're at rest. So let's set up our graph here. There's negative 70 right there. Negative 55. Why did I write that number? That's our threshold. So I'm going to put some dashed lines here. And that is our threshold line. Once the membrane potential reaches that threshold, then we have that all or none thing happening, and the active potential gets generated. I'm also going to put 0, and I'm going to put plus 30. Okay? At rest, all gates are shut. All the gates are closed. There's no movement of ions right now. And our resting membrane potential is negative 70. Okay, so we're going to do this I'll do it in red. So rest phase 1, we're right around here. So here's 1. We're still low. We're still around negative 70. Nothing's happening yet. So therefore, that's where we are. Everyone good so far? We're at phase 0, essentially, ground phase, resting phase. Nothing's happening. We're there. Now, let's go to phase 2. Now, phase 2, remember, as those sodium channels start opening, these are all voltage-gated channels. At this voltage, everything's closed. But as we start creeping up, because all these sodium channels don't open and close at the same time, some start opening up earlier, we start getting a gradual increase in our membrane potential, and we're getting towards threshold. So the next step that happens is depolarization. So step 2 is depolarization. So yesterday we talked about depolarization, and what ion is responsible for depolarization? Sodium. So sodium now has to move. So in phase 2, sodium gate is now open, and sodium can now come inside the cell. Because the sodium channel is open, let me put that little inactivation gate here, because that will come into play in a second. So there's a little inactivation gate. But the gates are open, so sodium can now move. So sodium is now coming inside the cell. At the same time, because we are not at the voltage where the potassium gates open yet, potassium is still closed. So there's no movement of potassium during depolarization. Just sodium is moving. And that causes the depolarization to occur. Everyone good so far with depolarization? Just sodium is moving, potassium gate is closed, because we're not at the voltage where these gates are going to open up. Okay, so let's go back to our graph. And we'll add in 2 is dipole. Depolarization. And it's just sodium flux. So sodium is moving, and that's it. Okay, so we'll get to phase 2 now. And we can see we reach threshold. Sodium channels are open, and we go all the way up. So all or none happens once we cross 55. Negative 55, we go straight up. All or none, all the channels open up now, because that's where the voltage-gated sodium channels like to be opened up, around negative 60. So they all open up, negative 55, boom, we have our extra potential. All the way up. Now, okay, that's step 2, depolarization. Just sodium is moving, nothing else. Step 3, after we depolarize, we have to repolarize. Okay? Let's make sure I'm right. Okay, so step 3, repolarization. We have to get the membrane back to resting potential. So we have to repolarize the cell. At this voltage, now we're up to 0 plus 10 millivolts. We're getting high because that's what we're showing right there in our graph with phase 1. Or phase 2 with depolarization, we went all the way up. So now we're at plus 30. Around plus 0, that positive, we get to 0, that's when the potassium gates now open. And when they open, what happens to the sodium gate, let me change the color. So the sodium gate is still open. However, during this voltage, that little inactivation gate now pops right in the middle, and it prevents sodium from coming in now. So there's no more sodium movement because in repolarization, the inactivation gate for sodium pops in the channel. No more sodium can move because that inactivation gate blocked the hole. Sodium can't go anymore. The door's still open, but there's an inactivation gate preventing flow of sodium. So there's no more sodium flow. But now we are in the voltage where the potassium channels can now open. So now potassium is wide open. So what's going to happen to potassium now? It's going to go out. So all these little potassiums inside the cell now have free action to leave the cell. Sodium is not moving, so that one positive charge is non-existent, it doesn't matter. But potassium is positively charged, and it is still moving. It's leaving the cell. So what's going to happen to the membrane potential of the cell when all this potassium leaves? It's going to go back down. And that is what repolarization is. Potassium movement only. That's what causes repolarization. That's why we're so very cautious of potassium concentration, because it can really screw up all these problems here. Yeah, question?
Does it stop at -70 and then it just all restarts?
We'll get there. So everything shuts off and shuts down. So we'll get to that in a second. So we get repolarization to occur. We're at this plus 30. So let's put phase 3 is repolarization. And this is only due to potassium flux. That's it. Repolarization is just potassium. AA, have you guys started EKG yet? Yep, okay. The T wave that you guys, AA, we'll do EKG in cardio when we get to that in 2 weeks. But I know AA already started, so I want to give them a little bit of a head start. The T wave represents repolarization of the heart. That's all the T wave is. So what ion is going to influence that T wave the most? Potassium. So if you look at any changes in potassium, whether it's too high or too low, you're going to see changes on the T wave in an EKG because that's what's going on in that phase. So the repolarization is the T wave. The depolarization is the QRS complex. That's the depolarization that's occurring. When you see the QRS complex, that's depolarization. When you see the P wave, that's atrial depolarization. So this whole thing is happening in the atria to cause the P wave. The QRS is ventricular depolarization. Repolarization is the T wave. So when you guys go through that, that's what we're learning here. So now we're repolarizing. So we're going to come back down. Okay? So we go back down, we repolarize. So we're coming back down because of potassium flux. Everyone good so far? The last phase is your hyperpolarization. What does hyperpolarization mean? Overcompensates too much, we go too low. So what's our goal? What should we drop down to? -70. So with hyperpolarization, we go a little bit lower than that. Yesterday we talked about, I said you need to know the numbers, but this is where it's coming into play now. So I said the Nernst equation, the equilibrium for potassium wants to get to like -90 when it flows by itself. So because the only ion that's moving right now is what? Potassium. So potassium, because it's the only thing that's moving, is going to want to go all the way down to -90. That's the equilibrium for potassium. So the hyperpolarization that we get goes a little bit lower, and then creeps back up again. That's the hyperpolarization in phase 4. And that's, again, due to potassium. You can put like overshoot. It went too much, not a big deal, because remember, these are voltage-gated channels. We're dropping the voltage. So as we drop that voltage, what do you think is going to happen to our potassium channels? They're going to shut down. They're going to close. They start to close. The sodium channel, now that it is coming down again, that inactivation gate is going to reset. So let's draw the blue doors for sodium. So now we are back to the doors being closed, and the inactivation gate has reset. Sodium potassium ATPase pump keeps going, resets all the concentrations, and this whole thing can start all over again for another active potential. So we're getting back to resting membrane potential. So what we did is we just drew it, and it just follows that way to this, to this, to that, and that cycle keeps going on and on and on. As long as you have a stimulus that's strong enough, you will get an active potential, and that will allow you to do whatever it is you need to do. So always remember, depolarization is due to flow of sodium. Repolarization is always due to flow of potassium. Okay? Resting, everything is closed. Sodium channels start to open up. Membrane potential starts to rise. We hit threshold. All sodium channels open up. Full depolarization due to sodium. As the voltage goes up, we hit the voltage gated requirement for potassium. Potassium doors open up. Sodium channel inactivates. And now we have just flow of potassium. When potassium is flowing by itself, we repolarize the cell, bringing it back down to baseline. We get a little bit of a hyperpolarization due to potassium going unopposed. And then all the doors reset, and we start over again. As you're studying, the way I talk it out, do it with yourself too. Just talk it out. Know the pathway, which way it goes. Any questions about generating of active potentials? Okay. So let's skip this stuff. Okay, cell communication. Cells have to communicate. Okay, we have nervous inputs that tell the muscles what to do. They have to communicate somehow. There are two major ways that we communicate. We have electrical synapses and chemical. Electrical synapse has gap junctions, which you see in cardiac muscle. These gap junctions allow the heart to beat as one muscle. There's a really, really great GRE word or SAT word that goes along with that. Anyone know what that's called when the whole heart beats as one unit? Starts with an S. It spells weird. It's called syncytium. Anyway, we'll get to that in cardio. But the whole heart beats as one because they're all have these gap junctions. The electrical conduction goes through the entire heart, and we get an active potential and the heart beats. Chemical synapses, they release these neurotransmitters. They change a electrical signal to a chemical one, and that chemical one can then act on the next nerve or muscle to cause another active potential so you can move your muscle, so you can breathe, think, whatever you want to do. So those are the two ways we have cell communication. Chemical synapses can be excitatory or inhibitory. So remember, we don't want things just to be stimulated all the time. We need control. That's why all of the majority of all of our feedbacks are all negative, to allow for control so things don't go rampant and go wild. So it can be excitatory or they can be inhibitory. Okay, skeletal muscle. So we're going to go through a little bit about muscle. We'll go through smooth muscle. We'll leave all the cardiac stuff when we get to cardiac next unit. So obviously the basic unit. Function of muscles to generate force of movement. we have electrical or chemical stimuli causes a mechanical response. Okay, I don't know what they do in high school anymore, but I remember when I taught high school, we used to have a lab where you have like the frog leg, and you put a little metal to it, and you stimulate it with electricity, and that muscle will twitch. That's a conversion of an electrical force to a mechanical force. In your bodies, when you're muscle, we have what's called a neuromuscular junction. The nerve secretes acetylcholine, which then activates the muscle fibers to contract. So it's a chemical conversion that causes a mechanical force. Okay, there are three types of muscle. Skeletal is striated and voluntary. Okay, so your skeletal muscle, if I say move your arm, you can move your arm. That's skeletal muscle. It is voluntary. Cardiac is also striated, but it's involuntary, which helps us out. We don't have to worry about thinking about having a heartbeat. It just does it automatically. Same thing with breathing for the most part. Breathing is mostly passive. We'll get to that. We get to pulmonary. So that's cardiac muscle. And finally, we have smooth muscle, non-striated, and involuntary. So smooth muscle, they cover the bronchus around your windpipe. All your vessels have smooth muscle in them, and it's how we control your blood pressure by altering the tonicity, the strength or the contraction or the dilation of those vessels. We can alter all that. So that's smooth muscle for different types. So here's our neuromuscular junction. Okay, so we have our spinal cord. We have this nerve that comes all the way down to the cell body, and then that nerve is going to attach itself to the muscle and innervate it to allow movement and activity to occur. So this is what the neuromuscular junction is, where they combine and come together. And we'll skip on to the next one. Okay, motor neurons, all of them are going to release acetylcholine as the neurotransmitter. So in our muscular system, it's always going to be acetylcholine is the neurotransmitter. A lot of the drugs that we give block acetylcholine. So you have what's called these cholinergic side effects. You'll learn about that later on, but everything I teach you, I know where you have to be to knowledge and where you have to go and why this is important. That's why I'm trying to bring in things that you will learn. Oh, this is why it makes sense. This is why I have to learn this. So acetylcholine, ACH, is the neurotransmitter, synthesized via the enzyme choline acetyltransferase. So this is what starts the process to make acetylcholine. And the amount of acetylcholine in a single presynaptic vesicle is called a quantum. So we have our little vesicles filled with acetylcholine, and the amount of acetylcholine in each one is called a quantum. That's all. We have postsynaptic membrane. So we have, this is where it's coming down, active potential comes down, and then we have the next neuron, muscle fiber, whatever it is, but this is called the postsynaptic, and this is your presynaptic. This has the vesicles. These have the receptors for it. Okay? Start getting in the habit of always associating what neurotransmitter you're talking about and what the receptors are. So start always associating the neurotransmitters with the receptors. When it comes to the neuromuscular junction, acetylcholine will always bind to nicotinic, with an N, nicotinic receptors. So just kind of get that in your brain now. Skeletal muscle releases acetylcholine as a neurotransmitter from the presynaptic neuron. It then binds to the postsynaptic side on the nicotinic receptors to promote the next, contraction of the muscle or whatever. So when these nicotinic receptors are bound, causes depolarization. Okay, depolarization, what ion's moving now? Sodium, it's depolarization. Sodium channel is moving. Okay? So Each quantum produces a small depolarization, but when you add all these little vesicles, the quantum of acetylcholine together, it's enough to produce a threshold and get above stimulus to cause a reaction to occur. So that's why it's called an excitatory postsynaptic potential. Yes?
Sorry, can you repeat the part where you said skeletal muscle releases?
Okay, so skeletal muscle, so the neuromuscular, the motor nerve will release acetylcholine. It will bind to nicotinic receptors on the postsynaptic side, which then causes the depolarization of that muscle to occur. And we get movement. A single motor nerve impulse produces an end plate potential that exceeds threshold, therefore you have movement. If you didn't have enough of a stimulus, you won't have an action potential generated, therefore there's not going to be no action to occur. So you need a proper stimulus to occur. Any leftover acetylcholine that hasn't been bound to a receptor, it's still kind of floating in that synaptic cleft, that little space in between, gets broken down by acetylcholinesterase. So anything that ends in ASC at the end is an enzyme. So start putting these things together. If they end in ASC, it's an enzyme. It's going to break down acetylcholine. And these little bits, the choline and the acetyl group, are brought back up by the presynaptic membrane, repackaged as acetylcholine, and is ready to go for the next stimulus that comes down the line. Okay, so the choline is taken up to make more acetylcholine. Now, some drugs and toxins that affect the neuromuscular junction. Botulism, okay, Botox. Okay, you're blocking acetylcholine release. If you don't release acetylcholine, is anything going to stimulate the muscle to contract? No. And that's why it works for wrinkles. It also is used for like people who sweat a lot. Okay, you can inject it at the area and all those sweat glands that have muscles to squeeze to produce the sweat all get paralyzed and you don't sweat as much. I think they're also using Botox for like headaches now and migraines. So there's only different things, but that's how it works. You block acetylcholine, it doesn't get released, nothing stimulates the postsynaptic membrane, and no action potential is generated, no movement. There's another drug we give called neostigmine. These won't be on your exam, but again, just reinforcing where your mind has to go now that you're in medical school. Start thinking about it in this way. So neostigmine is a drug that inhibits acetylcholinesterase in the synaptic cleft. So if I give you neostigmine, what do you think is going to happen to your acetylcholine in the cleft? Is it going to go up or is it going to go down? Why is it going to go up? You can't break it down. I blocked the enzyme that breaks down acetylcholine, so more of it will stick around in the synaptic cleft. Myasthenia gravis is a disease where we give this drug to. It allows acetylcholine to stick around longer so that you can still get your motor movement. Without that drug, they don't have enough acetylcholine to produce any type of movement, and they get paralysis in different areas. That's what happens with myasthenia gravis. And that's what I talk about here. So there's your myasthenia gravis. So our skeletal muscle is organized in a way in the things called sarcolemmas, and they help the sequence of events that influence development of movement. Ability of muscle to generate force when stimulated is due to our muscle proteins. We have proteins in our muscle, and they allow that to happen. So we have all these muscle columns. They're just cords, and when you bundle them all together, they get stronger. It's like if you have a piece of string by itself, it's very easy to break, but if you put a whole bunch of pieces of string together, wrap them up, they become stronger as a bundle. Same thing with your muscles. We have muscle bundles, and they're arranged in sarcolemmas. Muscle cells are multinucleated and bounded by the sarcolemma. That causes the striations, okay, because they have nucleus. That causes the striations. Smooth muscle doesn't have it, that's why you don't have striations. Each myocyte, so get your medical terms. Myo is what? Muscle. Cyte? Cell. Okay, muscle cell. Several myofibrils, which show light and dark bands under the microscope. So we have ways to look at them. We have dark bands and light bands. So each repeating pattern is called the sarcomere. This is the fundamental unit of contraction for all of the muscles. This is going to happen in one muscle fiber. But when you add them together to the thousands of muscle fibers that you have, then you have enough force to generate your movement and allows you to do things. The Z disc or the Z line is the boundary. That's where the Z lines are. We have two types of filaments. We have thin and we have thick filaments. The thin filament is called actin. And on this actin, it's got other proteins called tropomyosin and troponins. And we'll talk about that a lot more when we get to the cardiac muscle. We have thick filaments, which is the myosin. The thick filament does not move. All it does is it binds to the thin and grabs them and pulls them in. So the thin filaments are the one that actually moves. The thick filament, the myosin, is like an anchor, and it just sits there. And it provides an anchor for the thin filaments to move. We have A bands, which are the darker areas, and the I bands, which are the lighter ones, because the I bands don't have the actin there. So it shows up lighter. Okay? So the thin filaments have three major components. The backbone is the actual actin itself. And the way that it's made, we have this groove here. And that groove allows other proteins to kind of slide into that space. The main one is this tropomyosin, which has three troponin molecules on top. Troponin T, C, and I. T is the anchor. So T keeps the anchor tied to that tropomyosin. That's what the T troponin does. It's just an anchor. The C troponin binds to calcium. Remember I said yesterday, there are two things to remember with calcium. What are they? Contraction and exocytosis. So here's the contraction side of things when it comes to calcium and how it works. So the troponin C binds to the calcium. When troponin C binds to calcium, this troponin molecule moves and allows the binding site to be exposed. So as long as troponin C is bound to calcium, as long as you have calcium in the area, that troponin C will make the troponin molecule move out of the way, exposing the binding site that will allow the actin and the myosin to bridge, allowing you to have a contraction. Troponin I is inhibitory. So when there's no calcium around, troponin I kind of wins out and it inhibits contraction. We don't want constant contraction. We want to have the ability to relax the muscle. If you can't relax the muscle, then you're in constant contraction. That's called tetany. We'll get to that in a little bit later on when we get to neuro. So for now, for troponin, just remember, T is the anchor, C binds to calcium, and I is the inhibitory one to prevent the binding of the two. So we have the thick filaments, which are the myosin, and they help generate the force. So we have a myosin head, and that head will bind to the actin, but only when the binding site is available. And when is the binding site available? What has to be bound? Calcium bound to what? C troponin, the troponin C. As long as that's going to happen, that binding site's open, the myosin head can then promote that cross bridge to occur. Okay? So the heads are the cross bridges that bind to the actin and allow the movement to occur. Myosin heads are connected via tail, a little tail right there, and that allows them to bind to the thick myosin protein, that titin protein they call it, and that allows like the structural for the force to occur. So what happens, the thin filaments are being pulled over the thick filaments. So as an example, if Okay, so if this arm is this is the myosin, this is the titin protein, here's the little tail, here's the myosin head, here's the thin filament. All they do, I'll go this way. All it does, the myosin head grabs the actin and the wrist moves. That's the force, that's all it is. And then once we get to this part, calcium drops off. So what if calcium goes away, what troponin molecule takes over? I. When I takes over, they break. Then everything resets. And then as long as we have ATP available and calcium, the next cross bridge can occur and the next contraction can occur. So that's basically all that happens. In relaxed muscle, it's inhibited due to troponin I. That's the one predominantly. You're not using your muscles. You don't need it. When muscles are stimulated, intracellular calcium increased, binds to troponin C, and it exposes the active site. So now we can have a cross bridge form. Cross bridge to myosin, cross bridges to actin, performs a power stroke. That's the wrist moving. That's the power stroke that allows the contraction to occur. And then becomes cocked again, so the wrist moving back to the original position. We have to restart that cross bridge. So everything has to go back to normal position. That's what they're talking about here, where detaches, becomes cocked again, and then reattaches. That's all that they do. Grabs on, pulls it, grabs on, pulls it. When you relax, the fibers elongate back out to resting position, and they're ready to go again for the next contraction. This cycle will continue as long as there's calcium and ATP present. Okay, which is why when you have like rigor mortis, when people die, we're not producing any more ATP anymore. So that's when they get rigor mortis, they get locked in a certain position because there's no more ATP generated to relax that contraction and let those cross bridges detach. So that's why rigor mortis sets in afterwards. And as the thin filaments move in, the Z discs approach because the whole apparatus shrinks in. So the Z discs are the outside barriers. They start to contract, so the Z lines come together. So let's go through this step by step with the pictures. Step 1, sarcomere muscle before shortening, so that's where we are at rest. 2, low calcium, because there's no calcium, the active site is not exposed, there's no binding going to happen. And if you see, we have ATPs attached to that myosin head. It says ADP plus P right now, but it's still ATP. There's 3 phosphates there. We dissociated one because when this phosphate breaks off, that allows this whole action to occur. So step 2, low calcium. The sites are unavailable. Nothing is happening. Step 3, high calcium starts to come in. We got this influx of calcium. So it binds to the troponin, which molecule? C. And now that binding site is exposed. When this happens, we phosphorylate a phosphate off of that ATP. It now becomes ADP. When you do that, it causes energy to be released. And now, because of that phosphorylation, myosin is going to bind to the actin. That's the first part of ATP. We have to pop off that phosphate. That provides the cocking action so the myosin head can now attach to the binding site. Then, called the power stroke. When you have the power stroke, ADP, the whole thing now is released. And that is enough of a generation to cause the movement to occur in your power stroke. So now we actually have the contraction when the ADP is released. That causes the power stroke. So now we cause this contraction to occur. We are now contracting. We are in that power stroke. The next phase, ATP will come back and bind to that myosin head. And when it does that, it allows the energy to detach itself from that myosin crossbridge. Myosin actin crossbridge. So you need ATP back now to detach the actin and the myosin. That's why rigor mortis with death occurs. There's no ATP generated to come back onto that myosin head to detach the cross bridge. So you're stuck in that contracted position because you don't have any more ATP generation. That's rigor mortis. So this is the actual cycle. Calcium comes in, binds to the troponin, moves the troponin molecule to expose the actin binding site. Then the myosin head phosphorylates one of the phosphate groups off of the ATP, and we get binding of the actin and myosin. Then the ADP, which is left over, that bounces off, and that causes the power stroke to occur. Then ATP binds again to the myosin head. That allows for detachment of the cross bridge. And we can reset back to normal position. And as long as calcium is still there and ATP are there, you will have continuous cross bridge cycling. So when you're studying this skeletal muscle, make sure you understand how calcium is important and ATP is important. Where do they come into play in this cycle of contraction of the muscle? Yes?
Where is the cross bridge?
Between the actin and the myosin. That's the actual cross bridge. And then it dissociates with ATP coming back in. Okay. So the cross bridge is right here. See how they're attached? Yes.
In the picture in the slide before, would I be on?
Well, the whole, you see these they're like three molecules together?
Okay.
They're all three together. The C and the I and the T, they're all together.
And they just do different things.
Yep.
They're all on. Okay.
They're all in the same together, but when the C gets bound to calcium, it moves, exposing that binding site.
Okay. What would I be? What would it look like?
Just that when calcium goes away, the I kind of moves back into that spot.
On there, so it won't connect.
So the receptor site is blocked by I. It's gone. Mm-hmm. Okay, and then after sarcomere, after muscle shortening, remember the Z lines come together due to the contraction occurring. Okay. And we talked about rigor mortis already, so there you go. Okay, this last little bit, and then we'll stop here for a break. Excitation, contraction, coupling. This is a very, very important concept to understand. You could not have a contraction in the body without the cells being excited first. The cell has to depolarize, then the contraction occurs, called excitation, contraction, coupling. So, increased intracellular calcium causes concentration in all types of muscle. So let's look at the graph here. The first thing that happens, active potential in the T-tubule, don't worry about the T-tubule right now, just know that the active potential occurs. That has to happen first. This is the depolarization, right? This is the excitation, the active potential occurring. Once the active potential occurs, then we get an increase in calcium inside the cell. Then we get the muscle tension occurring. So you have to have the excitation first, then the contraction can occur. Excitation, contraction, coupling. So on your EKGs, when you're reading a P wave, the P wave is the atrial excitation. That's all you're looking at on an EKG. You know the next thing that's going to happen is the atrium is going to contract. When the QRS complex occurs and you have a ventricular depolarization, then you'll feel the beat afterwards. It's very fast, but the depolarization has to happen first, then you get the contraction. And what more do we have left? Yeah, let's take a break now. Come back at 1:25. Okay, let's get started. So, I want to mention a couple of things. For the AA students, when you look at the depolarization of a cardiac cell, it's different than what we've been learning right now. We only are learning about nerves and muscle and how their depolarization occurs with exponential with sodium and potassium gates, all that stuff. The cardiac excitation is different. We won't go into that till we get to cardio later on, but when you're studying, it's going to look different because of calcium in cardiac. So when you're looking at it, don't freak out like, oh, it's different. They're supposed to be different. So calcium. Where is calcium stored? We talked about yesterday, we talked about some pumps. We talked about one of the ATPase pumps, the calcium ATPase pump. The job of the calcium, well, the calcium is located outside the cell, and it comes in, but it also surrounds the sarcoplasmic reticulum, which is the contractile system. And this allows for a very short distance for diffusion. When you start extending the distance of diffusion, depending on how many layers it has to go between, diffusion slows down. Which is why people who have like bronchitis and they have all the emphysema and all the mucus in their lungs, they have problems with O2 is because diffusion distance is now increased from all the mucus that's there. That shouldn't be there. Yes?
What's SR?
Sarcoplasmic reticulum. Okay? So to return to that relaxed state, we have to get rid of the calcium, right? Because if the calcium is still in the cell, what's it going to bind to? Troponin C, which is going to cause what to occur? A contraction. But we have to relax. We have to allow the muscle bridges to break, reset, and go back to a resting state. So we need our calcium ATPase pump to pump calcium back into that sarcoplasmic reticulum. That's how we get rid of calcium to allow the muscle to relax. You have to pump calcium in. It causes the binding to troponin C, but then you have to remove that calcium as well. And that's where this calcium ATPase pump comes in, because you're pumping the calcium out and from inside the cell back into the SR, which technically is inside the cell, but it's sequestered. It's not floating around in the cytosol. It's sequestered in the SR. So technically, it's not intracellular calcium anymore because it's put into an apparatus, the SR. Okay, and stored calcium is weakly bound to a protein called calsequestrin, which will become more important when we get to smooth muscles. The force of the contraction is due to the firing pattern of the motor nerves in the muscle. You will only recruit the amount of muscle that you need to perform the action. For example, if I'm lifting up this water bottle, I'm only going to recruit enough muscle to overcome the resistance to lift this. What happens if, remember if you're going to pick up a bottle and you think it's full, so you grab with a lot of force, but it's empty and it goes flying? That's what we're trying to avoid. So your body only recruits whatever you need to do the activity that's required. That's called muscle recruitment. The way we get better contraction is temporal summation. What does temporal refer to? Time. So in this case, we start having rapid successions of action potentials or stimuli, and enough of those together will cause temporal summation. They all get added together to allow you to get above threshold so you can do your activity. At low frequencies, muscle generates force and then relaxes. So, okay, you're doing bicep curls. Okay, we generate a force and then we relax. We generate a force, then we relax. At high frequencies, the muscle doesn't have enough time to relax in between. So if you continue contractions faster and faster and faster, eventually the muscle cannot relax. There's not enough time in between for the muscle to relax. So you get a constant contraction called a tetanic contraction or tetany. Tetanus, you all had to get immunized for tetanus. Okay? what tetanus does, if you don't block it when it gets into the body, it causes tetanus. So you freeze up and you contract and you can't relax and you're stuck in a contracted position. That's what tetanus does. Okay, the force does not increase if the stimulation frequency is further increased because cross-bridge attachments are maxed out. So when you have tetany, all those cross bridges are used up and they don't contract anymore and they don't relax either. That's what tetany is. It's kind of like a frozen, contracted position. So you can see here are the stimuli. So every one is a stimuli. And you can see the force of contraction here. As they start getting closer and closer together, we get that temporal summation. And you can see here, the contractions are getting closer and closer, and the force is getting stronger and stronger because we have a lot of stimuli going on. But eventually, if it's too much stimuli, you get what's called summation, and you get tetany right there. That's what tetany is. And eventually, the stimuli starts to decrease again. And as it's decreasing, then the muscle will start to relax and go back down to normal. So I mentioned before that experiment in high school where you take the frog leg and you put a little electrode to it and you stimulate it. That's what they're doing here. You can send a stimulus, the frog leg will contract. If you increase the frequency of those stimulations to get to like something like that, the frog leg will seize up and it won't relax because you have too many constant stimuli. It causes tetany. Cross bridges can't relax, you're constricted and you can't relax. Yes? Is that like a muscle cramp? Not really a muscle cramp. I don't think it's a muscle cramp. It's more like if you get shocked by something, like seize up real bad. Like if you see people get tased, they start seizing like that. It's because you're stimulating the muscles and everything's contracting like crazy. So they seize up because the stimulation is so fast. So think of like a shock from like a stun gun. So I mentioned this before. If a greater force is needed, then we do what's called recruitment. We recruit more muscle fibers to allow what we do. That's where weight training comes into play. You're forcing yourself to recruit more muscle groups to overcome the weight that you want to lift. So you have to build muscle to overcome that resistance. That's what muscle training does. Okay, so we have this spatial summation. We talked about temporal before, that was time. What is spatial referring to? Space. Okay, just like Paul Atreides from Dune, right? Time and space. So in this case, we have one muscle fiber. So there's our muscle fiber. And we have multiple nerves that are all attached to that muscle to generate a bigger force. That's what spatial summation is. Now, this is sending an impulse, that's sending an impulse, and that is sending an impulse. We have 3 different nerves in space, all stimulating one muscle fiber. The temporal summation is one nerve stimulating that muscle fiber repeatedly faster and faster and faster. Or another way we can get to threshold is by having multiple nerves innervating one muscle. And all of those nerves stimulating one muscle group together will generate enough muscle fibers, recruit enough muscle fibers to overcome whatever force you need to do. Okay? Small motor neurons fire first, and they have very weak contractions. So you have a first little initial movement of your arm. You can't really generate much force over that, but as you continue the movement, then you get a lot more contraction. But that first little on the bottom there doesn't do much. Then you start getting those large motor neurons involved, which are less excitable, so they need a bigger stimulus to overcome threshold. But when it does do it, produces a very strong stimulus from the CNS to get a large motor group recruited to allow you to do whatever you need to do. So the small ones happen first, but they're very, very weak. Wait for that large neurons to kick in, and that'll kick in a bigger muscle group to allow a bigger contraction. Question?
Do spatial and temporal ever work in concert? Or
They do work together. Okay. And we'll go over that in a lot more detail when we get to the nervous stuff. And there's our tetanus toxoid we talked about before. So you see where that is. Now, within our skeletal muscle, we have different types of skeletal muscle. You may have heard this before, like your fast twitch and your slow twitch. What it comes down to is what do they use as an energy source, what's their energy metabolism, and so forth. So they differ, myosin and other proteins, and muscles will have different proportions of each, depending on what the purpose is. Slow twitch, or type 1, these are your postural muscles. You're all sitting in a chair right now, okay? Your vertebral muscles are keeping yourself upright while you're sitting in a chair. They don't fatigue because you guys have been sitting in a chair all day, right? You're not tired, but it's not like you're running a marathon and your muscles don't want to work anymore. So these slow twitch resist fatigue, maintain tone. Okay, the resisting fatigue is very important. You don't want to have to be able to like fall down all the time because you can't support yourself. We also have fast twitch, or type 2 muscle. These are like your extraocular movements of your eye. For you to move your eye around, your eyes dart around very, very fast. Why? Because they're fast twitch muscles. That's the whole point. You want to be able to see what's going around you, or if you hear a noise, your eyes will revert to that direction very fast because they're fast twitch muscles. Fast, brief movements, but they fatigue fast. Try moving your eyes back and forth real fast. After about 5 seconds, you can't do it anymore because they fatigue fast. They start hurting and so forth. That's what fast twitches are. Genetic differences. Sprinters will have more fast twitch muscles. Distance runners will have more slow twitch. So distance runners, more slow twitch, resist fatigue. They can go long distances further without lack of energy compared to other people who have more fast twitch. They're going to burn out faster. So here's a comparison. The color, slow twitch is red. They have a lot of myoglobin in there. Okay, fast twitch type 2 are white. They don't have a lot of myoglobin in there. Metabolism, slow twitch, oxidative, which means they use what as the source? Oxygen. Oxygen or aerobic metabolism is slow. It doesn't happen very fast. It takes a while to kick in. So slow twitch has very, very good oxidative metabolism. Fast twitch, glycolytic. They're using glucose as the initial energy source. When you first start running, that first 5 minutes suck because you're using glucose as your metabolism. You're using glycolytic anaerobic metabolism. Once that aerobic metabolism kicks in, and now you're ready to start using that, that's when you get your second wind, you get in that stride, and everything starts to feel okay when you're running because the oxidative metabolism finally kicked in and was able to catch up to the demand, and now you're in oxidative. Mitochondria, very, very abundant. Why? You got to use all this oxygen. Mitochondria is how you use oxygen to generate ATP, yes?
For patients who are declining, why do their lactic acid levels rise?
That I'm not sure. I can think about a couple of reasons, but I'm not actually sure why that lactic acid goes up on those patients. Maybe because they're just using more anaerobic metabolism, so they're generating a lot more CO2, which gives the lactic acid to go up as well. Yes?
Would the fact that there's more mitochondria in the slow twitch, would that be why they don't fatigue as fast too?
That I'm not sure of either, but the mitochondria provides all the ATP for them, so that's why they're able to resist the fatigue for so long, because they have all the great mitochondria to produce and to handle all the oxidative metabolism. You need the mitochondria to do it. So by having a lot of mitochondria, you can really oxidate a lot of, utilize oxygen a lot to make ATP. Glycogen content, how much sugar is there? Not very much in the slow twitch, but high in the fast twitch. You need a lot of glycogen, a lot of glucose reserves as your fuel source. So having glycogen stores very, very high in the fast twitch muscle makes sense. And the last one is fatigability. Low fatigability with slow twitch and high fatigability with fast twitch. Okay, some more muscle mechanics. There's 2 ways we move our muscles. First one, isometric. Iso means what? Metric length, or the distance, same length. So isometric, muscle develops tension but cannot shorten. What's an example of an isometric contraction? Pushing a door, a wall. You can also Push your hands together like this. This is an isometric contraction. My muscles are not shortening, but there is tension on my biceps. That's an isometric contraction. We also have isotonic. Okay, muscles able to shorten to carry a given load. So any type of bicep curl is an isotonic solution, isotonic contraction. Okay, fixed load, I put 5-pound dumbbell, I'm moving it up, that is an isotonic contraction. Length tension relationship. We, in our bodies through evolution many, many years, all of our muscles are already predetermined at the optimal length for our bodies. Okay, how is our optimal length fixed for us? Why is it fixed for muscles, skeletal muscle? What's all the skeletal muscle bound to? Bone. They are fixed. All your muscles are predetermined the length based off of the bony attachments that they are attached to. Make sense? So, we have to talk about a couple of things when it comes to contraction. These words will come up a lot when we get to cardio, but understand what they are right now. The first one, preload. What is the amount of force applied to the resting muscle before contraction or before tension? So I'm holding up this cup. Okay, this is the preload. It's a weight that's fixed. It's waiting to be done something with it. But this is the preload. Okay? That's what that's referring to. What is the weight before any tension is applied? So if you have a barbell or a dumbbell on your arm and it's weight resting at your waist right here, the tension that is on your arm is the preload. You haven't moved it yet, but it's preloading the muscle. That's the weight that's there. When muscle is stimulated, active tension is added to passive tension. And in the heart, we call that afterload. Example for that, very easy. This door, especially the front door to the building. You know when you push that door, first it's really heavy, you got to overcome that. When you first put your hand on the door and you push some resistance against it, that is the preload. Nothing's moving yet. You're waiting to push the door open. Okay? I have my hand on the door. There's some resistance, the preload in my bicep waiting to do something. Now I have to overcome the resistance of the door to push it open. That is the afterload. I'm adding the resistance to overcome that movement. So if you have a 5-pound dumbbell, to get it going, you have to apply more than 5 pounds of resistance or you can't overcome it to move the bicep, to move the dumbbell. That's afterload. You have to overcome the added weight to produce a contraction. And that's what the afterload is. So preload is what's on the muscle before contraction occurs. Afterload is what must be overcome, the pressure or the force to overcome to have a movement occur. That's what afterload is. So in this case, all we're talking about here is we have this optimal filament length, which is where Y is, and our body is already built to be at Y. If you're in these different ranges, whether you're all the way down in X or you're in the Z area, you can see here what it's pointing to. When you're in the X, I'll pull it up here. You see all this overlap? All that overlap is wasted actin that is not being bound by myosin, therefore it's not being a contractile, not being part of that contractile apparatus. So when the sarcomere length is too short, we get overlap of the filaments, weakening the contraction available to be made because now they're overlapped. If the sarcomere length is too long, too wide, then the proteins don't overlap as much anymore and you can't generate a contraction. So your contraction goes down. So that's why when you look at the big graph, the length of the sarcomere, the amount of force that you can generate goes down on both sides. But luckily for us, our bodies are already made at Y. So all of our muscles are already pre-made at the optimal length. But the reason why, because if it's too short or too long, the proteins don't overlap right, and you can't make a contraction. This is important for cardiac muscle. And this is what I was mentioning before about for the AA with the calcium. In cardiac muscle, as muscle length increases, the muscle becomes very sensitive to calcium. The heart doesn't have any fixed attachments. It just floats in your chest and just bounces around, right? There's no bone attachment I can attach to make my heart strength increase, but we get built in these things where if our length of our cardiac muscle increases, becomes more sensitive to calcium. Why is that important that it's more sensitive to calcium? What's the calcium going to do? Bind to what? Troponin C. And as you're studying, continue on that thread. Troponin C, which then moves the troponin molecule out of the way, releasing the binding site, which then can bind to the myosin head. Go along the whole way as you're studying. Okay, explain it out, talk it out. It'll make sense. So the length tension relationship I already talked about, we are already fixed. Our body is already made at the proper length test, so we're okay. Preload is not fixed in cardiac muscle, and we'll deal with that a lot more when we get to the cardiac stuff. So in clinical correlation, in a healthy heart, increased preload will make our myocytes stretch, which makes a more forceful contraction. Okay. In a diseased heart, cardiac muscle can become very stiff and reducing the ability to stretch. So therefore, you have problems there. So that's an example of what happened with the heart, but that won't be on your test for sure. Our force-velocity relationship, resting muscle length is set by a certain preload. Whatever it is that's on your muscle before you do anything, that's the length of your muscle. So if you're starting here, this is the length of my muscle. If I'm starting down at the bottom, obviously my bicep is a lot longer than it was up here. But the length of that muscle is already predetermined the minute you add a preload to it. The weight a muscle must lift after the resting length is established is called that afterload. That's that extra weight or resistance or pressure you have to generate to overcome that preload. The speed of shortening decreases as the total load increases. If you had a 1 pound barbell in your hand, you can go real crazy. Okay, now go 50 pounds, you're like struggling to get one, whatever it is, The heavier the weight, the slower you can move. Okay, that's all that's referring to there. So the speed of a bicep curl, 1 pound dumbbell versus a 50 pound dumbbell, which one's gonna move faster? The lighter weight one. However, in cardiac muscle, increased force and speed of contraction can be produced without changing preload because the heart has what's called contractility. And we'll get to that a lot later when we get to the cardiac stuff. Smooth muscle lines the walls of most of your hollow organs, so your vascular system, your GI system, respiratory. If you have asthma, you know about smooth muscle contraction very well, because with asthma, the smooth muscle contracts around the bronchus, causing narrowing of the airway, among other things, but that's one of the examples. So smooth muscle is a very important therapeutic target. A lot of the medicines that you guys are going to be giving are going to attack the smooth muscle. That's where a lot of the regulation occurs. So homeostasis is disrupted by too much or too little smooth muscle tone. If you have excess smooth muscle contraction, respiratory tract, that's your asthma. Another example of a lack of vascular smooth muscle tone is during septic shock. In this case, all those vessels are wide open. There's nothing that's holding them tight or constricted. All the vessels dilate, your pressure drops. That's why they have septic shock and their blood pressure drops like crazy. There's no more tone telling that smooth muscle to contract and maintain your pressure in your system. So that's just an example of having too much or too little tone. They're not striated because they're not organized into sarcomeres as well. They do have thick filaments and thin filaments, but they're not set up in a nice line anymore. They are kind of like all over the place in a different array. And what that does, it allows, is think about your stomach. if you think about how a snake moves, in order to get all the food stuff, everything through your intestine has to squeeze in a certain weird way to propel all that food going forward. So it kind of squeezes and pushes forward like a snake does, getting through like the water or whatever. That's what our smooth muscle does, and that's why it's set up the way it is, to allow churning, movement of the food, whatever else got to be. Also allows for a greater force over a long period of preload. So we can have this force for a long period of time with the way that's made up. There's two types of smooth muscle. We have visceral smooth muscle, which is the most common type, large bundles. Here's that syncytium again, that word. That has to do with gap junctions, electrical activity, fast conduction through the entire system, allows it to function as one unit. And we have multi-unit smooth muscles. They don't have any gap junctions. A lot of autonomic neuron supply. So your autonomic nervous system is your sympathetic and your parasympathetic systems. And fine control over force development. So like in the iris, so with your diameter of your pupil, your smooth muscle is involved with all that stuff as well. You guys in PA today have the lights out, right? You learn all that stuff. We're going to go through all that in neural as far as like what you're really looking for. But that is smooth muscle reaction to you adding a stimulus. Excitation, contraction, coupling is here as well. Obviously, it's a little bit different in smooth muscle. So in smooth muscle, we have intracellular calcium going up. Supposed to happen, right? Just like in skeletal muscle, goes up as well due to these voltage-gated calcium channels. Smooth muscle does not contain troponins. So it doesn't have that type of molecule where the calcium binds to the troponin C. It doesn't have it. So what happens in smooth muscle, it forms this calcium calmodulin complex. When this calcium calmodulin complex is activated, it causes myosin light chain kinase to phosphorylate, and that causes the active myosin to occur. Okay, so we have this calcium calmodulin complex, activates myosin light chain kinase, which activates the myosin, so now we can have a contraction. Now, when calcium is decreased, remember we have to have relaxation. We can't just have constant contraction all the time. In order to relax, we have to get rid of the calcium. So when calcium decreases, muscle relaxes due to activity of myosin light chain phosphatase. And that will relax the smooth muscle. So when you're studying this, myosin light chain kinase is for activation, contraction. Myosin light chain phosphatase is for relaxation. There is no troponin C moving out of the way, actin, binding, all that fun stuff. That's only in skeletal muscle. Smooth muscle has calcium calmodulin complexes, activates myosin light chain kinase, causes contraction to occur. When calcium drops, myosin light chain phosphatase comes out and deactivates the system so you can have relaxation. So when you're studying this, just know those two. Sphincters. We have a lot of sphincters. They help us out a lot by allowing us not to evacuate ourselves at times we don't want to. So we have sphincters. They have to be contracted for a long period of time and maintain that force over long periods of time. So the way they can do it without high rates of ATP is because they have what's called a latch bridge state. And that latch bridge state allows them to be contracted and connected for long periods of time without the need for constant ATP. Remember with skeletal muscle, in order for that muscle to relax, you have to have ATP come back, bind to that myosin head to allow it to detach from the actin. So you constantly need ATP a lot for skeletal muscle. Smooth muscle doesn't need all that constant ATP because of this latch bridge state. It allows it to have that constant tension and tone to allow you to have that sphincter tone there and not go away. Okay, we are done now with general phys. Come back at 2:10, give you 15 minutes, and we'll start Neuro.